In this video, we will continue our discussion about SARS-CoV-2 and ACE2 glycosylation study. So, let's just understand first about SARS-CoV-2 as spike protein trimers, glycosylation, uh, characterization. So, in order to understand the glycans that are present on SARS-CoV-2 as trimer, they uh, cleaved off N-glycans with the help of PNGase treatment. So what they did it is treat this SARS-CoV-2 spike protein trimer with PNGase treatment. After that, they permethylate these glycans so that uh, they can easily be ionized in the mass spectrometry, and uh, signal strength will be better compared to non-permethylated glycans. And next thing that they did for O-glycan analysis, they used beta elimination method. So these two methods they have used to analyze N-glycans on O-glycans. And briefly, what they have identified is that in the spike protein trimer, they have identified 49 N-glycans and 15 O-glycans were found. And in order to determine the occupancy, that is how frequently these sites were glycosylated, they used sequence deglycosylation strategy, which involve endoglycosidase H and PNGase F in the treatment in the presence of 18OH2O after tryptic digestion of S. So we are not going right now into the detail of all of this process. Our main focus of this video is to understand the result. So what they have find out is that uh, asparagine at 1173 and asparagine at 1193 has low occupancy compared to other N-glycan site. Other N-glycan site has efficiency greater than around 95%, whereas these two N-glycans has efficiency of 52% and 82% respectively. And in order to understand the micro heterogeneity of the SARS-CoV-2 S trimer, what they have done, instead of treating this uh, as spike protein with single protease, that is trypsin, they treat it with three different proteases so that they can have more peptides and more better glycoprotein fragmentation they can observe in the mass spectrometry. And in this they have identified that complex glycan of the SARS-CoV-2 spike protein are under process, mainly particularly they are under galactosylation. The galactosylation and silation are below than what has been observed in the normal human cells. However, the core fucosylation is high compared to human cells. And oh, this is wrong. It is not N-linked. It is sulfated glycans. There are small amount of sulfated N-linked glycan. Small percent of sulfated. Instead of this small, it is sulfated N-linked glycan are also found in SARS-CoV-2 spike protein. So this is a representation how the data has been presented in the paper, and this is the overview. In this we can see that around more than 25%, just like around 30% are high mannose glycans, and less 20 around 20% are hybrid, and most of them are complex structure. And this is in-depth information about what kind of structure are they, whether they are bisected, biantennary, triantennary, tetraantennary. So we can see that from our previous slides we came to know that core fucosylation is high, so we can see that fucosylated glycans are higher in the analysis. And this color also represent the terminal end of this glycan. in this case, and we can also see the terminal end on the in this glycan structure on the top of this bar graph, and here also the what does that mean is also represented. So for example, this bisected has the N terminal of galactose, whereas this biantennary has most of the biantennaries has the and this terminal end or non-reducing end. Yeah, that word is correct. In this bisected, the terminal or non-reducing end is galactose, whereas in biantennary it is glycneck, terminal end or non-reducing end is glycnet, and so on. And this one is different. This is leg die neck is multi can be either silated, fucosylated, or it can be either silated, fucosylated, or galactosylated, galactosylation. So it can be a mixture of any of these three. And this is an example that has been presented in the paper to show how they have find out the micro heterogeneity. So they have done a spectral count of each type of glycan that they have find out on a specific site. So this is an example of like in the spike protein of SARS-CoV-2, as trimer of SARS-CoV-2 asparagine at position 74, and usually the counting is done from N-terminal side, and they have found that these different kind of glycans can be found in on at this site. However, the most abundant one is this one and these three one, and it's depend on what kind of analysis one is doing. But we can see that most abundant glycans are this one 30, 29, 28, and same is the example, and similarly, and the same spectral count has been done at different sites, such as this asparagine at 149, asparagine at 0801, asparagine at 0234. We can see that in 74 and 149 the spectral count and the spectral count is on the higher side compared to this panel number D and panel number E. From this, a general idea can be made that E and D has low occupancy, or asparagine at 234 and 0801 has low occupancy compared to 74 and 149, and that we have and this this result we have already uh, known, we are already aware of, and this result are already been aware, or we are already been aware from the previous slides. So that's how they have done some statistics calculation. This is not in depth about how they have what cutoff they have used to consider whether it is a high abundance or low abundance. These things can be available in the paper, and the link of the paper is available in the description. Thank you for your time. Have a good one.